But whilst we're waiting, I'll give you a bit of a background on who I am and introduce myself. So I'm Dr. Nicole Hellessey. I'm a new postdoctoral researcher in Professor Mark Weisberg's lab in the School of Biology at Georgia Tech. But I've previously done my bachelor's in marine science, a master's of Antarctic science, and my PhD in Antarctic science, all at the University of Tasmania. So the seminar today is actually based more on my PhD research than on the postdoc. So if you have any questions about the research that I'm currently doing at Georgia Tech, I'd be more than happy to answer questions about it at the end. But for the time being, I'll be talking mostly about the PhD research that I did whilst I was at the University of Tasmania. I was particularly lucky that my PhD was actually done in conjunction with a number of organisations. The major ones are shown on the title slide here, so the Commonwealth Scientific and Industrial Research Organisation, or CSIRO, the Australian Antarctic Division, where we use their aquariums, the Institute of Marine and Antarctic Studies, Aker Biomarine, which is a krill fishing company, and the Antarctic Climate and Ecosystems Cooperative Research Centre. So I was incredibly lucky to be part of a large-scale research project as part of my PhD, and it was a multinational and obviously a multi-institution project. So hopefully people will be able to continue joining in whilst we get into it. So to give you a bit of background, obviously my main area of focus is Antarctica. And I know that most people in the Northern Hemisphere would be more familiar probably with the Arctic and the Northern Hemisphere and the North Pole. So to give you a bit of a background, particularly of Antarctica, not only is it the polar opposite from the Arctic in that it's the South Pole, but Antarctica is a frozen landmass surrounded by ocean, whereas obviously the Arctic is a frozen ocean surrounded by land. That causes a few major differences in the ecosystems between the two. The prominent ones being that the northern, in the Arctic, you get more terrestrial and land-based animals in the ecosystem. So you get polar bears, you get wolves and caribou, Arctic foxes and rabbits. You obviously still get the marine life up there, so you get walruses and narwhals, whereas within Antarctica, the entire ecosystem is based around that marine sphere. In fact, there's only one really true terrestrial animal that lives in Antarctica, and it's an insect, a midge. Every other animal that lives in Antarctica has at least some part of their life cycle or requires the marine element of the environment for their diet, which was incredibly essential for my research looking at krill. So I focused on Antarctic krill, which is a particular species known as Euphorsia superba. They're about 6 centimeters long, and they are really well known for having this bright green digestive gland, which is generally green because of all of the algae that they consume. And they also have a stomach, which is actually in front of that digestive gland. It's a very, very small area just before that larger green digestive gland. And most of the dietary analysis that's done on krill is either gut analysis, using broken pieces of what can be found in the gut under a light microscope, or lipid analysis, so looking at fatty acids and the oils within the krill. I primarily focused on the second one. So krill's diet is driven predominantly through the environment in Antarctica, which of course goes through huge seasonal fluctuations. So at the top we have Antarctica during the austral summer, so February, and you can see that the sea ice is incredibly contracted during the summer. And because of that, you have a different environment within that ocean because there isn't sea ice impeding things like wind, mixing that water. You also get more sunlight going into the water. You also get warmer water being in the summer, and you don't get as much stratification within the water. Whereas in winter, when there's more sea ice, it almost doubles the size of Antarctica, and it also then stops that mixing of the water from the wind. You get more stratification with underneath the sea ice, And of course, you also get cooler waters and you get less sunlight below where the sea ice is. So you get two different types of diet depending upon where the krill is. The krill feed mostly on plankton, and in the summer, the most common type of plankton is phytoplankton. Phytoplankton is just algae. It's probably more commonly referred to as just algae. The two main sources of phytoplankton that they consume is diatoms and dinoflagellates. And depending upon which one they're eating, you will see different dietary markers within their lipids and their fatty acids. During the winter though, with less sun and cooler waters and that stratification, you do still get some phytoplankton on the bottom of the sea ice, but it's certainly not as much as what you would get during the summer months with those larger algal blooms from the spring melt of all the sea ice. So they are still eating plankton, but it's a different kind of plankton. It's zooplankton, zooplankton just being animal plankton. So this is a copepod, which is a large component of krill's diets throughout the winter. They also eat fish eggs, squid eggs, small larvae, bacteria, marine snow. They're filter feeders, so they'll eat pretty much anything that can stick to their feeding webs. So krill are at the basis not only of the ecosystem in Antarctica, which is a wasp-wasted ecosystem where krill are that pinch point within the food web itself, but they also form the basis of a fishery within the Southern Ocean. So we were lucky enough to work with an industry partner, which was a company called Aker BioMarine based in Norway, but they fish for krill in Antarctica. And there's three very distinct fishing regions within the Southern Ocean, which are highlighted here in red for the South Atlantic, a bluey purple for the Indian, and green for the Pacific. I was lucky enough to go down to Antarctica twice with my PhD. The first time to Casey Station, which is right on that border between the Indian Basin and the Pacific Basin, and the second time to the West Antarctic Peninsula, below the tip of Chile and Argentina in the South Atlantic. The majority of the fishery, 97% of the krill catch, comes from that South Atlantic region, particularly those lighter pink areas. So we'll be focusing mostly on those, as that's where the fishery was and that's where our samples were collected. We were incredibly lucky that Aker BioMarine collected 100 krill for us each day that they were harvesting for 3 years. It was an incredibly long-term continuous data set that had never been done before, particularly in that region, but we've yet to find a study in other areas that is that long and that continuous for Antarctic krill in particular. We were also lucky enough to get some samples from the K-axis voyage, which went to the Kerguelen Plateau during 2016 with the Australian Antarctic Division. So initially, one section of my PhD was going to look at a East Antarctica versus West Antarctica comparison of krill diets. But we were then lucky enough to have a opportunistic sampling of krill from near the Bellini Islands in the Pacific Ocean, or the Pacific Basin, with the Antarctic Circumpolar Expedition and a researcher called Giuseppe Malvo. So we then decided that we'd actually look at the differences between the ocean basins surrounding Antarctica as part of my PhD. So we were incredibly lucky for that opportunistic opportunity. So the fishery for krill mostly focuses on two different things that they turn krill into. One is probably more commonly known than the other. So the more common one that people know of the krill is used for is omega-3s and krill oil. So they are found in most chemists and pharmacies around the world. They are one of the largest and fastest growing nutraceuticals. The research on krill oil is incredibly diverse and is getting more funding, and there's more clinical trials happening every year. The other one is actually livestock feed, and it's one of the ones that people don't tend to think about very much. So krill was actually used as a substitute for proteins, particularly in things like beef, sheep, and pigs. And then once they found that they could not only take the omega-3s and the proteins of the krill, but they could also take that astaxanthin that gives krill that sort of lovely, rich, reddy color and feed it to salmon, and it would make the salmon predominantly pink. or even more pink in the case of salmon where they're naturally occurring, they would substitute in krill into the livestock feed in the aquaculture to give salmon that beautiful bright pink colour that we all know. My research was predominantly focused on the omega-3s, obviously focusing on those fats and lipids that like to work their way up through the ecosystem. And in particular, the industry wanted to know how the omega-3s were changing and how they were differing, particularly with the environment. So krill, because they are that center of the ecosystem and they feed all of those larger sort of charismatic animals that we think of when we think of Antarctica, a lot of the nutrition and obviously all of their energy and their lipids come from feeding on krill. So the lipids that krill have are not only incredibly important for the ecosystem, but also for the fishery. So whilst we were working with the industry, with the exception of them collecting the samples for us, there was only really one question that they had, which was entirely unrelated to the questions that I had for my PhD. So the two main questions that I wanted to ask as part of my PhD research was, how does krill's oils change in time and space, and how do they change with the environment? So we had ourselves a bit of a 1 in a quillion opportunity, if you'll excuse the pun. So we were not only able to get a large continuous data set of krill harvested for us from the fishery, which is unheard of in terms of scientific research of krill, because mostly if you are doing krill research, you'll be going down during the spring and summer seasons to Antarctica. You'll be competing for logistics within the ships and on the bases. And obviously funding is a huge reason why we can't really stay down and get as many samples or go to as many places as what we'd like to be able to do krill research. So having an industry partner where their entire job is to go and follow the krill swarms and they can collect them for us throughout the entire year, it really was an opportunity that was a bit too good to miss. So the way that our samples were collected was a little bit different to most normal krill fishing vessels. So most vessels would have a harvesting bag with a large plastic cod end on the end, a normal sort of trawl bag. Aker BioMarine has a patented system, which is more of a vacuum system. So they actually have a long hose that connects to their ships and then has a small bag attached to the end of it. The krill get sucked in when the pump is turned on, and they come up within the water in that hose. The hose then opens obviously at the other end onto the boat, and the water flushes away and the krill are left intact and in really good condition. So you don't get as much cod end feeding using this system, which means that you don't get as much carnivorism within the krill's diet, because krill will eat each other under levels of high stress. Which means that you don't actually pick up as much carnivorism within the diet as what you would using other methods of collection. Obviously doing this, we also get a really diverse range of samples of krill, so we get different ages, different sexes, different locations, and different environmental conditions that they're harvesting the krill in. So we were very lucky that they were able to collect everything from their GPS location, the wind direction, the depth that they were collecting the krill at, right through to what the ambient air pressure was. So switching gears just slightly, I'd like to give you a little bit more background on lipids and oils in general, just because we're going to be delving into particular fatty acid research, and it can get a little bit complex very quickly, particularly because there is a lot of acronyms, and the structures within the diagrams and the results will be a little bit hard to interpret without that background. So fatty acids are the shortest amount of the carbon base chain of the lipids, so they can join onto structural backbones. So those larger backbones are lipid classes, things like triacylglycerol, which is a storage fat. It's generally what we think of as our belly fat. And also phospholipids, which are structural fats. So they are the fats that make up the membrane in your cell membranes. Fatty acids then join onto these backbones, and the most common ones are the ones that we think of as biomarkers, if you've ever heard that term before. And the three most important ones, particularly when it comes to krill and omega-3s, are SDA, EPA, and DHA. So EPA is 20, 5 with 3, DHA is 22, 6 with 3, and SDA is 18, 4 with 3. So if you see those numbers coming up in the figures and the results, I've also put the acronyms there to try and help you, but that's the fatty acids that we're talking about. So SDA in particular, that 18, 4 with 3, is one of the things that the industry was asking us and questioning us about, because the specifications of the monograph that they used to make krill oil omega-3 tablets didn't have SDA in it, but it could fluctuate within the krill's fatty acid profiles between being 0 and up to 14% of their fatty acid profile. So on occasion, not just Aker BioMarine, but other krill fishing companies would have tens of thousands of litres of omega-3 oil ready to make into krill oil tablets, only to test it and find it doesn't meet specifications for what krill oil is, and would then have to throw out those tens of thousands of litres of omega-3 oils or potentially sell them as something other than a krill oil. So they wanted to know where this SDA was coming from and how they could potentially change the monograph. It gets a little bit complicated though, because the biomarkers and the fatty acids vary throughout the body. So as I mentioned earlier, krill not only have a stomach, but also that digestive gland. Krill generally don't store any lipids or fats within their stomach. Their stomach is an initial processing site for them to start digesting any prey that they may have gotten through their filter feeding. Whereas their digestive gland does hold a lot of lipids, and it's where they store a lot of their food until they're able to metabolize it. So even between two different parts of their digestive system, there's huge variations. You also get a lot more omega-3s, such as EPA and DHA, within things like krill's eyes, as it helps lubricate their lenses when they're at like incredible depths and under a lot of pressure. You also see variations in these fatty acids and biomarkers in different life stages of krill. So gravid females, which are full of eggs before they spawn, are much higher in lipids and oils in general, as they are more prominent in the eggs before they are spawned, as those oils then become the nutritional basis of what the krill larvae will live on as they undergo metamorphosis, coming back up to the surface as they spawn on the sea floor. They come back up to the surface of the ocean. They aren't able to eat or find nutrition with filter feeders until they get closer to the surface, when algae starts to become more prominent and they go through their last metamorphic stages. So that lipid in those oils is incredibly important to those early life stages. Is the more lipids they can pack into those eggs, the more likely those early life stages will survive. Similarly, you'd get less lipids, less oils in things like males, as generally the males are seen as not needing as much lipid to be able to reproduce or do what they need to do to survive within the environment, whereas females are the ones that generally are seen migrating much further distances out to the continental shelf, and that's because that's where they spawn. So they have not only much larger swimming distances that they undergo during their life, but they also go through those huge variations with their egg production. So you can get a more detailed gut analysis using things from within the stomach and putting them under a microscope. So generally speaking, gut analysis is a little bit biased, as it underrepresents things like soft bodies, so things like fish eggs and squid eggs, obviously bacteria and viruses, marine snow that krill may be taking in will be underrepresented in any gut analysis. However, the things that do have hard structures, so things like diatoms and dinoflagellates, even if you're lucky enough to find pieces, they may have been crushed and therefore hard to ID. They might have been partially digested, and you may not be able to find what you need to actually get a good identification of what species you're looking at under the microscope. The one benefit of the lipid biomarkers is that even though they are very broad and general categories, you do actually get a much better overview of everything that the krill has eaten, including those soft bodies. So getting into the results now. So we have the total lipids. So this is all lipids, all oils for the entire body of the krill in milligrams per gram of dry weight of the krill. So this is scaled for the size of the krill. So if you have a krill that is incredibly small but really, really fat, it will be scaled against a krill that is very, very large but much leaner. And then across the x-axis, we have the different years of collection and the locations that they were harvested from. So the acronyms are WAP for the West Antarctic Peninsula, SOI for the South Orkney Islands, and SG for South Georgia. And you'll be able to see pretty quickly that the fishery migrates between the West Antarctic Peninsula and the South Orkneys, which it tends to flip between during the summer and autumn, and South Georgia, where it stays during all of the winter into that early spring. Unfortunately, there aren't a lot of spring samples from the fishery, as that's actually the time of year that they go and do all the maintenance on their vessels, and it's also when they do all the production of their krill oil tablets and that aquaculture feed. However, it joins really nicely with the data sets that we already have from scientific voyages and from scientists on bases. So we're actually able to fill in that gap between that spring and summer using data from other studies. But this was the first study to have continuous data throughout the entire summer, the entire autumn, the entire winter, and the beginning of the spring. And the first thing that was really apparent to us was this sinusoidal nature of these lipids. They increase during the summer and autumn to their highs, and then decrease across the winter into their spring lows. Now, this had always been hypothesized, but no one had ever actually shown that this was what the fatty acids and the lipids were doing within krill. So this was an amazing takeaway for us just to begin with before even delving any deeper. So going to the next level into what krill were eating, we can break down these total oils and these total lipids into those lipid classes. So if you remember, I was talking about triacylglycerols. So that's your belly fat. That's in the bright sort of yellow. And the phospholipids, those structural fats that make up things like your cell membranes, is in that light blue. And as you can see, krill are mostly using those two lipid classes. And you can see that that storage fat, that belly fat, fluctuates in the same way that the total overall lipids do. So again, increasing through the summer and autumn, and decreasing again during the winter and the spring. Now it gets slightly more complicated at this point, as most krill, whether they are Arctic, Antarctic, or even the ones in the Gulf of Mexico, generally use wax esters as their main form of lipid storage. However, Antarctic krill, the specific species of Antarctic krill called Euphorsia superba, don't use wax esters. As you can see, that red at the bottom of those bars is incredibly small. They generally tend to store their fats as triacylglycerols, but it gets more complicated again, as krill are also known to use phospholipids as a storage. They keep it in an emulsion within their first thoracic part of their body. So it's an incredibly complex story, which is why we have to break it down even further from those lipid classes. So this is a principal components analysis, or a PCA plot, and you'll see a few of these, so I'll just go into a bit of an explanation of how to read them. So a PCA plot will take all of the data that you give it of various inputs, and it will try to decide which ones are most similar to each other. The more similar things are to each other, the closer they are to each other in the graph, and the more distinct they are, the more that they are pushed further away, and the longer those little sticks will go out to what that particular variable is. So you can see quite clearly in this particular diagram, which is using the fats and the omega-3s from the whole krill, so this is using every part of the krill, including their shells, we can see that the Indian Ocean is distinctly different to the Pacific and the Atlantic. We see that krill within the Indian Oceans are distinctly different between themselves at different sites. So we have Indian Ocean Site 6, which is predominantly seeing high levels of DHA, that 22,6 with 3, and some EPA, although at a lower level. We're seeing, though, that those krill from the Atlantic and from the Pacific basins are incredibly high in SDA, though, that 18,4 with 3, which, if you remember, is the fatty acid that the fishery was having such issues with when trying to meet the monographs of what they're making their krill oil tablets with. And then we have the other two Indian Ocean sites, 4 and 5, which are a little bit off by themselves. And they're generally higher in diatom markers, 16,1 with 7C, and with copepod isomers. So that's the 21 with 9C and 22,1 with 9C. So already you can start to see that there's these differences between the more diatomaceous SDA and DHA on one side, and the more diatomaceous but also more carnivorous side with the Indian Ocean. Taking it one step further, we can look just at the fatty acids within the stomach. Unfortunately, krill stomachs, because they are so light on in having lipids, because they very quickly pass into the digestive gland and then get stored there instead. Unfortunately, we had to pull stomachs between the sites. So the 12 different samples that we had at Indian Site 6, we then had to pull into 2 stomach samples, as we had to combine 6 stomachs into a single sample to get enough lipid to actually get a reading. So all of these samples have 6 stomachs per sample, just to make sure that we were consistent with how many krill stomachs were going into each one. However, that then gives us another issue, that obviously some krill may have just recently fed, whereas others may have not eaten directly before being harvested. However, we can see that there is really close correlation between these sites, so that gives us really good hope that there is a strong confidence in what we're seeing, that this is more true to their diet. And you can see again, the diatoms are more towards Indian sites 4 and 5, although to a lesser degree. Indian Site 6, again, very high in DHA as well as in EPA, although the EPA is almost halfway between where Indian 6 and that Pacific site is. And you can see the SDA again starting to lean more towards the Atlantic and the Pacific. But you can see that the Atlantic in particular is incredibly high in 14 naught, which is alpha-linoleic acid. And that can sometimes be used as a marker of where things sit within the trophic food web. So generally 14 naught accumulates as you go up the trophic system. So potentially krill in the Atlantic are actually sitting at a slightly higher trophic level than what we see in krill in other ocean basins, which was something that we weren't expecting to see. Diving a little deeper once more, we then look at the lipids within the digestive gland. And again, these were pooled, but we only had to pull 2 digestive glands per sample, so you will get 6 points for each of the sites here, as opposed to the 2 seen for the stomachs. So each one of these dots represents one sample which has the 2 digestive glands within it. And again, you see that Indian sites 4 and 5 are far more diatomaceous. We get more copepods within Indian 6, but interestingly, we see more copepods at one Atlantic site than the other within their digestive glands. We also see that all of those major omega-3s, SDA, EPA, DHA, all incredibly closely linked to each other, all sitting, nestling next to each other within that PCA plot, and all of them sitting right between that border between the Pacific and the Atlantic. So from this, we can already start to tell that the omega-3s, particularly within the digestive glands, are far less common within Indian krill, and that may be because they're metabolizing them straight into their bodies. They're not holding them within their digestive gland, they're metabolizing them straight into their whole bodies. But it may also be a case that there are so much algae because there is a more consistent level of algae in the background environment within the Atlantic and the Pacific. The krill within these ocean basins are able to keep much higher levels consistently within their digestive gland, even though we don't see that within their stomachs. Keep in mind that Indian Site 6 was the one that was very high in DHA, both in the whole krill body and in their stomachs, but we see them much lower here in their digestive gland. which really does hint towards that they're metabolizing these as quickly as they can, as we aren't seeing any in their digestive tract, but we're seeing incredibly high levels within the bodies themselves. Take it one step further, this is the same PCA plot of the whole krill. So you've actually seen this graph before, but instead of labeling it with the oceans and the sites, they're now labeled with the oceans and what particular sex and maturity stage those krill are. And you can already start to pull apart that there are differences within the sites. So with the Atlantic and the Pacific, you can see that all those whole krill are really tightly grouped together. But you can see that things like Atlantic males are more similar to each other than they are to gravid females. So those big, nice, beefy, full of eggs female krill. You can also see when you look at the Indian sites that subadult females, those open diamonds, are far more similar to each other than they are to those spent females. So once they've spawned all those eggs. And you might be thinking, but if this is the same diagram we've seen before, we know that those are sites 4 and 5 on the right-hand side of the PCA and site 6 on the left-hand side. And you're absolutely right. We couldn't actually find any spent females at sites 4 and 5, nor could we find any subadult females at site 6. So unfortunately, there is a bit of a bias here as well, in that we don't have subadult females from site 6, we don't have spent females from site 4 and 5, which unfortunately, it means that there is some sort of entanglement between site 6 having that high DHA and the spent females also having high DHA in the Indian Ocean, and we can't really disentangle them. So there is a lot of potential to go back, particularly to the Indian Ocean, and see if we can untangle those in future studies. There's also a lot of potential to go into the Atlantic and the Pacific again. As we can see, they're closely grouped together, but there are a few krill within those samples that we can start to tease apart, that there are some that tend to waver more towards copepods, and there are some that tend to waver more towards those dinoflagellates, particularly the ones that produce that SDA. So depending upon where these krill have been harvested from, we may actually be able to find, well, why was it that some krill were having far more copepods within the Atlantic at one area than at another? So we've looked at a bit of spatial difference, and we've looked at a bit of temporal difference with those different seasons and those different years. So to add in the last little sort of smattering of what we looked at, there's the environment. So to do this, I used satellite data. The two main types of data that I used were ocean color, which is a proxy for chlorophyll A. So you take the wavelengths of red, green, and blue, and then the ratios between them. You can actually determine if the ocean is slightly more green or slightly more red, and you can use that to decipher if there's chlorophyll A present in the water. And that is then, of course, a proxy for chlorophyll A and that primary production. And we also use sea surface temperature, which of course is incredibly important, particularly in the Antarctic, as things start to warm up and we start to lose sea ice. We were able to match these satellite data using the GPS locations of the fishing vessel, as we knew that obviously krill were there at that location at that time because they were harvested for us at that location at that time, which meant that between the GPS locations of the satellite data and the GPS locations of the fishing vessel, we could then match those data to each other. Unfortunately, chlorophyll A, that ocean color data, is incredibly hard to get within the Southern Ocean. There's a few issues which I'll go into later. So the data itself was interpolated across a 3x3 kilometer grid, with the central point of that 3x3 grid being the location of the fishing vessel. And it was also interpolated over 1 to 8 days. So if we were able to get an exact value for chlorophyll A on the day that they were harvested, that was what we preferred to use as our data and would keep in our data set for analysis. If we weren't able to get data on that exact day, we then opened it up to any data within that 3 by 3 kilometre area within 8 days of the krill fishing vessel being there. So that fourth day within the 8 is the day that the fishing vessel was actually present. So it's incredibly hard to get satellite data in the Southern Ocean, and that's for a number of reasons. Part of it is cloud cover. The Southern Ocean is very cloudy. Doesn't matter what season it is, it's always very cloudy when you go south. Part of it is the sun angle. So satellites are more built to go around the equator and temperate regions. Unfortunately, people didn't really think about polar regions, particularly with satellites, for a very long time. And it means that the angle is far too steep when you get to that tip and that sort of apex point of where the poles are within Antarctica and even the Arctic as well. So it's incredibly hard to get data with the sun angle to get colour. You also have issues with things like sea ice and the albedo effect, so that reflection off the ice coming back with light. So sometimes, even if you can get a clear sunny day in the Southern Ocean to be able to get your ocean colour data, you will be foiled at the last hurdle by sea ice being in the way. So it's incredibly hard to get that ocean colour data, in particular. Sea surface temperature, less so, because it can be done through a laser. Whereas the ocean colour, needing either a picture or a photograph to be able to then get that colour data from, or direct light wavelengths being reflected back to the satellite. So having a look at the 3 different areas within just the South Atlantic, so we didn't use the samples from either the Indian or the Pacific for this, and it's because those samples were only taken from one time point, and we only had one particular sample really from the Pacific because it was opportunistic. And even in the Indian, we only had 3 sites and they were taken over the course of a fortnight. So we decided that it was better to put all of our effort into where that long-term data was coming from in the South Atlantic with the fishery. So all of the environmental and satellite data that we analysed came from that area. So the West Antarctic Peninsula, you can see here that the total lipid dry weight, TLDW on the left, chlorophyll A in the center, and sea surface temperature in degrees Celsius on the right. And you can see that there are a few patches where there are incredibly high levels of that total lipid within the krill, within the Gerlache Strait, that big open channel of water between the tip of Antarctica, the main continental part of Antarctica, and those islands slightly to its north. You can see that that area reasonably relates to where there's also high levels of chlorophyll. And again, you can see that it's an area where there are occasional days of higher sea surface temperature, which may be due to upwelling. Unfortunately, there's not a whole lot of other details that we can draw out of this, other than that there is one spot right towards the very southern end of this map, where there is a group of points of krill that were harvested, where they don't have particularly high total lipid, although there are levels of high chlorophyll and particularly high sea surface temperatures in that area, but we don't see that converting into higher lipids within the krill. The hypothesis, of course, being that with the higher sea surface temperatures, there should be more primary production, and that higher primary production would cause higher lipids within the krill. Moving slightly further north within the Atlantic, you have the South Orkney Islands. This is predominantly where the fishery was in the autumn. And again, you can see that the lipids are generally quite high, keeping in mind that the lipids are high during the autumn. That's when they're at their max before they then spawn and also then start going into using their lipids for storage and using them as their source of main nutrition throughout the winter as they start to decrease. So we see those really high total lipids across the board in the South Orkneys. There's a few areas of higher chlorophyll, but the sea surface temperature is pretty consistent across all points. And the reason the sea surface temperature is pretty consistent, even across those 3 years within our study, is because there is a canyon to the north of the South Orkney Islands that causes a dramatic level of upwelling. And it's already a known oceanographic feature, and it does mean that there is a small patch higher sea surface temperatures that you see regardless of the time of year because of that upwelling. However, the small top end of where that canyon is, I guess, you can see those few points of higher chlorophyll right in the middle of where the harvesting of the krill were collected. And those few points actually match up really well with where that tip of the canyon is. So potentially that initial point where the water comes up first is where the primary production is highest, but it then quickly disperses throughout the ocean surrounding the South Orkney Islands. And due to that, we see that the lipids are pretty consistent around the rest of that region. And lastly, we have South Georgia, so this is even further north, and this is also where the fishery was during the winter. So keeping that in mind, of course, we see that the lipids are much lower here within the krill because this is as they're decreasing those lipids across the winter and they're using them up, using all those fat stores. So we also see lower chlorophyll, which is to be expected during the winter months when there's more sea ice, the water is cooler. Obviously, you're getting more stratification, there's less wind, all those kinds of fun things. And then also you see the sea surface temperature. There's the occasional point that drops to 1, maybe negative degrees if you're lucky. But generally speaking, not as warm as what we were seeing in the previous samples. It's very hard, though, to show this data in a way that both represents the time component and the spatial component. It's easy to just show the spatial component in a map. It's easy to show the time component in an XY graph. It's very hard to combine the two. So this is the best attempt that we can. So you have chlorophyll in the green, sea surface temperature in the black, total lipids in the light blue, and EPA as a representative of what fatty acids are doing within the krill body in orange. And you can see that whilst there is some variations within the year, the general trends are the same across all 3 years. So we can see that in the summer and the autumn, when the fishery is within the West Antarctic Peninsula and the South Orkney Islands, as the temperature decreases, their lipids increase in general, so those total lipids increase. But we see a decrease with their fatty acids, particularly those omega-3s such as EPA. Then during the winter, when they move up to South Georgia, which is why you get that large temperature jump between the end of autumn and the start of winter, they go up to South Georgia where it's slightly warmer, slightly further north. But again, during the winter, as you see the temperatures decreasing, you see the total lipids decreasing as they're using up those fat stores. But we're actually seeing those omega-3 levels starting to increase, despite the fact that we know that the chlorophyll and the levels of algae in the water are much lower. Unfortunately, because of how sporadic and how hard it was to capture the chlorophyll data, we only really got to record one algal bloom in its entirety, which you can see in the first year in 2014. We were hoping, obviously, to have this in each of the years. Unfortunately, it didn't happen, but you do get small spikes at the start of the other 2 years. So we can hypothesize that there were algal blooms at those times. We just managed to miss them due to cloud cover, ice cover, or any other potential reasons. However, using that first year as an example sort of year, we see what we'd always kind of hoped to see. The sea surface temperature increases. That then causes the chlorophyll and that primary production to increase. We then see an increase in the lipids with a slight lag, with them metabolizing them into their bodies. And then once those lipids have reached a point where they no longer need to be storing them and using them directly, they can start to increase those omega-3s again, which was really exciting. That was what we'd kind of hoped we would see. Obviously, it's unfortunate that we only really got to see it in the 1 year, not in all 3. But the really good part here is that the project's actually continuing even now, even though I've now left. They now have a postdoctoral researcher who's working on it at the moment. So they've extended the grant out for another 3 years. So we're hoping that we'll be able to see some longer term trends, not just in krill lipids, but potentially in sea surface temperatures and in how those fatty acids are changing longer term as well. So there's obviously a few gaps and a few biases within the data, even as well as what we could do. So in the future, there's a lot of possibilities to actually go back and expand on this particular study. So one idea would be to do some year-round sampling of krill at the same location. Obviously, this is sort of pie in the sky thinking, because generally either scientists are on station or on voyages during the spring and the summer, or you'll be down over the winter season, but then it becomes much harder to get into those open water areas, particularly during the winter, especially if you go further south. There have been some studies that have done winter sampling of krill, though, particularly at Palmer Station, one of the US bases of the West Antarctic Peninsula. Christian Rees and Jen Walsh with NOAA have done some stuff with winter samples looking at differences in different life stages of krill in their fatty acids and their lipids. This would actually be a really wonderful little grant to put together to do a comparison between our samples of winter with South Georgia and the Palmer samples being further south. There's the potential that we could even do them on the same days and the same times as each other in the future, potentially. But definitely using past samples with the data that they already have and the data that we already have, there is a really nice little data project potentially in the future as well. Also year-round sampling, but in different ocean basins. Unfortunately, the regional study and that regional aspect looking at the different ocean basins was only able to be done on those summer samples. And that's because, again, that K-axis voyage and that ACE voyage into the Indian and the Pacific basins were only during that summer season, the normal time for scientists to sort of go down there. So we don't actually have year-round samples in those other seasons from those locations. So this was already a huge multinational and industry collaboration just to get the data that we already have. But obviously being able to do that on a longer time frame over potentially a whole year or multiple years would require very high level and large scale multinational and industry level grants and collaborations to be able to do it. The one good thing that we can take away from this, though, is that the Chinese krill fishery is starting to expand in the Indian Ocean. There used to be a much larger krill fishery in the Indian Ocean, but it actually shut down in the late 80s. They're starting to go back and sample, and they're exploring at the moment the fishery in the Indian Ocean to see whether it would be viable to go back there at the moment. But if they were, that would potentially give us an option to go back and say, if you're happy to collect 100 krill for us each day on your vessels in the same way that Aker BioMarine were, and keep them and store them for us and ship them so that we can analyze them, potentially we could get something from the Indian Basin and from the Atlantic Basin where we both have long-term samples. Of course, that then just leaves the Pacific, which is hopefully not far behind, particularly with that large MPA that they've now put in place in Antarctica. Of course, we also had issues with our satellite data. So in the future, it would be really lovely to be able to collect things like the sea surface temperature and the levels of chlorophyll at the time of collecting the krill. Unfortunately, whilst there were fisheries observers on the boat, they were recording things like the size, the sex, and the age of the krill that were being collected. They weren't actually trained or allowed to collect things like the sea surface temperature, which would have obviously really helped us with ground truthing our satellites. But it also would have been lovely to have gotten a few vials of the water at the time that they collected the krill so that we could see what algae was actually in the water at the time, so that we could actually get some much better ground truthing of the chlorophyll A levels as well. Obviously, we did get some direct environmental conditions from the ships. They kept logs of the depth and the wind direction and wind speed, the air pressure, ambient air temperature, and a few other environmental conditions. But at this point in time, we're not really sure how to include those and whether they're actually relevant to what's happening with krill and krill's diet. But again, there is a lot of potential there to go back and maybe reanalyze this data using those once we know what they could actually be prospectively used for. At this point, I'd like to acknowledge all of my wonderful PhD supervisors, Patty Virtue, Peter Nichols, So Kawaguchi, Steve Nicol, and Nils Holm. They were incredibly useful in teaching me not just more about krill, as my master's was actually on Antarctic fur seals. I'd never worked on krill before. But they also taught me how to do all of the lipid analysis, as I'd never done that before my PhD as well. So I'd love to thank them for their help with all the studies. Particular honourable mentions to Robert Johnson from the Bureau of Meteorology here in Tasmania. He got all of my satellite data for me and particularly helped me with analysing that. And also to Jessica Erickson, who was my lab partner throughout my PhD and my absolute rock. Also thanks to Aker Biomarine. Without them, obviously, we wouldn't have had our krill samples, let alone much of a PhD. So they get an amazing shout out as well. And also obviously to the Institute of Marine and Antarctic Studies and the Australian Research Council who funded the PhDs. Thank you so much for listening. If you've got any questions about krill diet, their life cycles, anything like that, I would absolutely love to answer some questions for the next 10 to 15 minutes. So I'll actually stop sharing my screen and hopefully we can get into some questions and answers.
Great presentation, Nicole. Can you hear me? This is Jeanette.
Yes, I can.
I really like what that you shared with us, your activities in the Antarctic. I'm really interested in how people make krill oil because the copepod that I work on also is full of omega-3 wax esters. But my question is, do you notice whether the decline in the lipids corresponds to the production of eggs? Because that's what my copepod, Euphausia antarctica, does. They build and store lipids as wax esters during the spring and summer, and then during the winter, they convert the wax esters into triacylglycerides, which is a more labile lipid, which is put into the eggs to be used for development. So the copepod reproduces in the winter. The eggs are nearly pure TAGs, while the adults are 70% wax esters. So my question is, does the decline in lipids correspond with egg production in the krill?
So the short answer is no. So Antarctic krill spawn in the middle of their autumn, so it's generally around April, May. Yeah. And obviously the winter in Antarctica starts in the June. So there's a couple of months between when they're spawning. And we also see that those lipids continue to be at those high levels right until the end of those autumn and even for those first few weeks of the winter.
Mm-hmm. Okay.
So, yeah, so unfortunately it doesn't quite correspond with when they're spawning, but we do know that the lipids do drop, obviously in spent females once they've released all of their eggs. We know that they do drop, which is why you'll see a little bit of variation in that those autumn lipids right at the top of that sinusoidal pattern. There's a lot more variation, and that's because there's those spent females in there as well that helps drag that down. But so thank you for your question.
So do you know what kind of lipid is in the eggs? Is it
predominantly it's sterols that they move into their eggs, a few triacylglycerides, but predominantly it's sterols because it's it's the most easily transportable around the krill body.
Yeah. Okay, thank you so much, Nicole.
Not a problem. Are there any other questions?
A question.
Yep.
Hi, Nicole. Great talk. So you mentioned that krill supplements sold at pharmacies are expected to have a specific lipid content, but that this content varies with diet and a lot of additional factors. Based on your work and possibly the work of others, do we now know what is the optimal diet to get these ratios?
So the monograph that the fisheries use to actually make their omega-3 tablets, it's built around and it's based off what they knew of fatty acids of krill at the time that the monograph was made, which was in the late 60s. So the monograph says, you're allowed between 20% and 40% EPA, you're allowed between 15 and 20% DHA, and you're allowed, 1 to 2% SDA and, a handful of other, fatty acids sort of thing. So obviously that large fluctuation in the SDA, which can go from being 0% to 14%, is what's throwing that out. So we already know that the dinoflagellates is what's causing that SDA within the krill. So we know that krill that are harvested after a diatom bloom within the ocean won't have as much SDA. But we also don't know if krill are retaining that SDA or not. That's something that we're still actually looking into. There's no real answer to that question quite yet. But we do also know that they've now changed the monograph based on this research. So there's a paper by Jessica Erickson, my lab partner that I mentioned at the end in the acknowledgements there. Her paper from 2018 has now actually gone on to be part of a case that they put before the Therapeutic Goods Administration here in Australia. And it then went before the FDA, so the Food and Drugs Administration within America. They have now changed that monograph based on this research. So it's now allowing up to 7% SDA. So still not as high as what the 14% is, but there's a lot more flexibility than the 0 to 2% than what they used to have. So they're no longer having to throw out anywhere near as many of their krill oils as what they did. And the ones that do have that high SDA, they can now dilute with other krill oils that they have that maybe might not be as high in the SDA to bring it actually into specifications before they then go into production. So we're actually really happy that we were not only able to answer the industry's question of where this was coming from, which is the dinoflagellates, but we could also help them so that they're no longer having to go out and harvest all of these thousands of krill to turn into omega-3 tablets, only for them to be thrown out again. So it's a really nice sort of scientific industry collaboration that we got to do some really amazing research, and we actually got to have a practical application for it as well.
Very cool, thank you. Yeah, it's really great when you get to apply your research in real time at the industrial level.
Yeah, it's a very exciting thing when you get told, you've actually made a noticeable and something that you can see difference in the real world, even if it's not something that I particularly think of each day. It's definitely a really cool thing to be like, I was a part of that. Is there anyone else? So I can see that there's a question in the chat there from Emily. So hi. Saying, are we able to link the high levels of SDAs to any particular foods other than dinoflagellates? We don't know of any particular species of dinoflagellate yet. There has been a bit of research on different species of algae within the Southern Ocean and in particular the West Antarctic Peninsula, but we don't know which particular species it is that the krill are honing in on and getting that SDA from. So that would be something really interesting to go into in the future. I know Peter Nichols, who was one of my supervisors with my PhD, he did his PhD way back in the day, looking at different lipids within the algae in Antarctica. So he in particular is very keen to get someone down there and actually take some samples and see if we can find which species is causing that peak in that SDA.
Very cool. Thank you for answering.
That's all right.